I want to give a visual, just a really quick visual kind of drawing that shows what's going on with DNA replication versus DNA transcription, the first step of protein synthesis, right? So there's two reasons why a DNA molecule will unzip, right? So first of all, DNA replication. So a DNA molecule will, sorry for my squiggly lines there we go. It will unzip, and there's a bunch of enzymes that do that. And then you have an exposed template, right, on both sides. And let's say that this is the 3, and this would be the 3 and the 5. Right. Okay, so this is replication. So this would be under what circumstance would the DNA replication take place? What time in a cell's life would you be replicating your DNA? During the synthesis or S phase, right, of interphase. And because you know that in the near future you're going to be dividing and you need an extra copy to send to each new cells, right? So during interphase, during the S phase, in preparing for eventual cell division, that's the purpose of that, to replicate. All right, so let me change colors here. Okay, so if you recall, and because everybody seems to get confused about this, if we're putting new DNA nucleotides, right? DNA nucleotides on both sides, aren't we? Right? And which way is that happen? Which side is the leading strand? Would the leading strand be the one above or the one below? Remember, are we going the one, oh, we're having some disagreement. Okay, so the answer is the one above would be the leading strand. You would be able to put new DNA nucleotides, right, going this way towards the replication fork continuously, because you always, the polymerases, no matter which polymerase it is, okay, polymerases read the template in a 3 to 5 direction. They read the template in a 3 to 5 direction. They create the complement in a 5 to 3 direction, right? So then this is the leading strand because it can go continuously. It doesn't have to stop as this keeps opening up here, right? And this would be the lagging strand that gets done in pieces because it has to keep going back and starting again, going back and starting again. So this would be lagging, right? All right. And in both cases, the new nucleotides, the new individual nucleotides, right, both here and here, whatever they are DNA nucleotides, right? Meaning that they would have A's and T's and C's and G's meeting up with whatever is here and here and here, right? Okay. All right. So hopefully that reviews that a little bit. I didn't go over the other enzymes because I was more concerned with people understanding the 3 to 5 direction seems to confuse people, and leading versus lagging. All right. Now with DNA transcription, what we're going to look at today, right? If you're transcribing Also, the reason why it only moves in a 3 to 5 direction has to do with the fact that polymerases are enzymes, and they have active sites that can only fit onto the DNA molecule if they're going in a 3 to 5 direction. I don't quite know how to describe that more in detail, but it has to do with the shape of the active site and the way that an enzyme is very picky about whether it's going to meet up with the phosphate facing towards it or away from it. I think is to do with the phosphate position on the 3.
Why can't it just start from both ends?
Well, because, I mean, if I go back up here for a second, these are good questions. I'm glad you're asking them. Okay. So the reason why is because you see here is 3 to 5, right? Right here is going 3 to 5 because this is 3 and then way out here is 5. The problem is that this replication fork here, right? This replication fork is exposing the DNAs going this way. So it's unzipping, 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 right? It's only going one way. And therefore, the enzyme only has access to the new free nucleotides a bit at a time. That's why. So that's why it has to keep the DNA polymerase has to keep going in and going up, back stitching, up, back stitching. In fact, Mr. Anderson described it as back stitching, right? That's why. Because the replication fork goes this way, and the DNA polymerase only gets access to the free nucleotides a little at a time and has to keep going backwards. Good question. Good question. Okay, so now we're doing transcription, which remember is the first step of protein synthesis. So this is a totally different circumstance. This is the first part of protein synthesis, right? So this happens throughout interphase, right? Throughout whatever the cell is doing in its normal life, it always, every cell, no matter what kind of cell it is, has to be making important proteins because proteins are the worker molecules of any cell. Like if you're a pancreas cell, you're spending your interphase making insulin, which is a protein, right? Which is very, very important to a body. So think of it that way. So all during interphase, right? Right? Because those proteins are the worker molecules of cell physiology and the cell can't function without them. So in transcription, again, you're unzipping the molecule. There we go. So again, we're unzipping the molecule, right? But now, we're not replicating both sides. We only need a little bit of the DNA. We don't need all of it. And in fact, we only need one side of the DNA that's exposed. And what the RNA polymerase is the same as the DNA polymerase in that it can only move along the strand in a 3 to 5 direction, right? So since this is your 3 to 5 direction here, this is going to be your DNA template, which is another word for guide. Template strand is down here, right? This would be the non-template. This won't be used, not during transcription, because it's going the wrong way, right? Okay, so along comes that RNA polymerase, and let's just put a few bases down here to guide it. Let's say that we've got an A and a T and an A and a G and a G and a C or something like that, okay? So the question is, the RNA polymerase, the RNA poly enzyme that's coming along here, right, is looking for the right RNA nucleotides this time, RNA nucleotides. So the RNA nucleotides, remember, are going to be a little bit different. There are going to be A's, there are going to be G's. The little T's I'm drawing are the rest of the thing. There's going to be C's and there's going to be U's. right? These are the pieces. These are the free RNA nucleotides that the RNA polymerase is looking for to attach in the right places, right? You notice there are no thymines because RNA doesn't have thymine. So RNA poly is going to put what here with the A? What's it going to put there? Yes, very good. Uracil will now go with A. Very good. And what will go with T? An A will go with T. Yes. And then with another A, you'll have a U. And then with G, C, G, G, and C, you'll have C, C. You got it. Yes. Okay. And it will continue going this way and adding nucleotides until it has a finished RNA transcript, right? Okay. So I just wanted to, and once again, the RNA polymerase, just like the DNA polymerase, can only read in a 3 to 5 direction, right? And can only build in a 5 to 3 direction. Just wanted to make sure that that was clear to everyone, because I think that will help when you go in and you do the rest of what you need to do today. And I'm not going to go into translation because that you'll get that from something else, and that's not really the comparison I was looking at here. But in terms of how the DNA molecule is used in these two processes, where in both cases you need to unzip the DNA and then use it for something, either to make more of it or to make a transcript so you can make a single protein, right? That's what's going on. Okay, excellent questions on that. I'm going to close this down.